Hello everyone. Let's discuss about vitamin B9 or folic acid groups. It is a water soluble vitamin. Synonyms are liver lactobacillus casei factor, liver lactobacillus casei factor, then vitamin M, then streptococcus lactis R factor or SLR factor. Streptococcus lactis R factor, then vitamin BC, then fermentation residue factor, then Pteroyl glutamic acid, These are the synonyms of vitamin B9. This is the structure of vitamin B9. It contains a pteridine nucleus, a pyrimidine and a pyrazine ring, that is pteridine nucleus. Pyrimidine and pyrazine ring, that is a pteridine nucleus. Pteridine nucleus. Then it contains a PABA, that is para-aminobenzoic acid in its structure. Para-aminobenzoic acid. These together form pteroyl or pteroic acid. Then it also contains a glutamic acid residue. So, pteridine nucleus, that is pyrimidine and pyrazine rings, then para-aminobenzoic acid and one glutamic acid. This together forms the entire vitamin B9 structure. Chemically, it is pteroyl glutamic acid. Pteroyl glutamic acid or PGA. Three compounds are termed as pteroyl glutamates. These are monoglutamate, triglutamate and heptaglutamate. These three compounds differ only in the number of glutamic acid residues. First one is monoglutamate. As the name indicates, it contains only one glutamic acid residue. It is synonymous with vitamin BC. Next one is triglutamate. Triglutamate means it contains three glutamate residues. This is designated as fermentation factor. This is fermentation factor. Next one is heptaglutamate. Glutamate. Hepta means 7. So it contains 7 glutamate residues. And it is synonymous with vitamin BC conjugate of yeast. This pteroyl glutamic acid is liberated from these conjugates by the enzyme conjugases. Next we will see biological active forms. Biological active forms. This active form is reduce the tetrahydro derivative, that is tetrahydrofolate or FH4. This is tetrahydrofolate. That is the active form. It is obtained by addition of 4 hydrogens to the pteridine moiety at 5, 6, 7, 8 positions. We will see the addition of these hydrogens to the pteridine moiety. That is the structure. Here NH2, NOH, then here comes NH, then here CH2, NHR, here H, then here also HH, then here NH. This is FH4, that is 5, 6, 7, 8. Tetrahydrofolate. We will number here 1, 2, 3, 4. This is 5, 6, 7, 8. So this tetrahydrofolate is 5, 6, 7, 8 tetrahydrofolate. That is here 5, 6, 7, 8 hydrogen atoms to the pteridine moiety. These are present mainly in the form of N5-methyl or N5-formyl derivatives. N5-methyl or N5-formyl derivatives. We will see the formation of FH4. Formation of FH4. This folic acid is first reduced to 7,8-dihydrofolic acid. Folic acid or folate is reduced to 7,8-dihydrofolic acid or FH2 or H2 folate by the enzyme reductase. Here one NADPH plus H plus NADP plus NADPH plus H plus NADP plus. Here one molecule of vitamin C. This is the first step. In the second step, this H2 folate or FH2 is converted to H4 folate or FH4 by the enzyme reductase. These two steps are catalyzed by the same enzyme. Here NADPH plus H plus NADP plus NADPH plus H plus NADP plus. Here also one molecule of vitamin C. So folic acid is first reduced to 7,8-dihydrofolic acid or FH2 or H2 folate and then to 5,6,7,8 tetrahydrofolate or FH4. Both the reactions are catalyzed by the same enzyme folic acid reductase, which use NADPH as the hydrogen donor. It requires a cofactor, that is very important, that is vitamin C. That is the formation of FH4. Next we will see the clinical importance. Clinical importance. Some of folic acid deficiency anemias may be due to inherited deficiency of the enzyme folic acid reductase. There is another one is folinic acid. Folinic acid. So what is folinic acid? This is one of the active form of folic acid. This is a formyl derivative. Active form of folic acid and is a formyl derivative. It is reduced tetrahydrofolate with a formyl group on position 5. Reduced tetrahydrofolate. Reduced tetrahydrofolate with a formyl group on position 5. So we can represent it as F5. So formyl group in position 5, FH4. This is a tetrahydrofolate with a formyl group in position 5. That is folinic acid. It is an active form of folic acid. This folic acid, when added to liver slices, it is converted to formyl derivative in presence of NADPH. Here vitamin C enhances the activity. This folinic acid is termed as citrovorum factor. Citrovorum factor. This is folinic acid. Structure of folinic acid is similar to that of folic acid. Difference is that it is the reduced tetrahydro form and with a formyl group at position 5. It is also called as leucovorin. Leucovorin. It is a reduced form and it contains a formyl group in position 5. It is also known as leucovorin. Next one is rhizopterin. Rhizopterin. A similar compound but with formyl group in 10th position. Here formyl group in 5th position. Here it is in 10th position, that is 5,10FH4. F10FH4. Next we will see the conversion of this F5 to F10FH4. Conversion of this F5 to F10. F5 to F10. First the F5 FH2 FH4 is converted to F510FH4. This is the intermediate. 510FH4. This is then converted to F10FH4. First step is by the enzyme isomerase. Here one ATP, ADP plus Pi, Mg plus plus. And the second reaction is catalyzed by the enzyme cyclohydrase. Cyclohydrase. So one molecule of water. This is a conversion of F5 to F10 FH4. That means F5 can be converted to F10 by the action of an enzyme system 4-methyl tetrahydrofolate isomerase. 4-methyl tetrahydrofolate isomerase in presence of ATP. At first, F510FH4 is formed. So, first reaction F510FH4 is formed. Then by the action of the enzyme cyclohydrolase, it is converted to F10FH4. There is another enzyme, 4-methyl tetrahydrofolate synthetase. Here it is isomerase. Then one more enzyme is there, synthetase. It is in pigeon liver and in bacteria. This enzyme catalyzes the addition of formate. Formate means HCOOH. It catalyzes the addition of HCOOH to FH4. This enzyme is specific for formate only. That is FH4 by the enzyme 4-methyl FH4 synthetase. One molecule of formate. Here also ATP, ADP plus Pi. It forms F10FH4. F10FH4, but this is in pigeon liver and in bacteria. Next we will see the formation of N5-methyl FH4 from F510FH4. N5-methyl FH4 from F510FH4. F510FH4. F510FH4 can be converted to N5-methyl FH4. It can be converted to N5-methyl FH4 by an NAD plus dependent reductase. NAD plus dependent reductase, and this CH3 group is then transferred to deoxyadenosylbutol. Deoxyadenosylbutol. It is cobamide coenzyme. Cobamide coenzyme. So, this F510FH4 can be converted to N5-methyl FH4 by an enzyme NAD plus dependent reductase. And the CH3 group is transferred to this deoxyadenosylbutol or cobamide enzyme, and it forms methyl butol. Methyl group is transferred to deoxyadenosylbutol and it is formed methyl butol. This is an important donor of CH3 group as occurs in methylation of homocysteine to form methionine. Homocysteine to methionine. That is the formation of N5-methyl FH4 from F510FH4. Next we will see the biosynthesis of vitamin B9. Biosynthesis. Many microorganisms can synthesize the folic acid. Some of them cannot synthesize this para-amino benzoic acid, which has to be supplied in the diet. In presence of ATP and CoA SH, PABA reacts with glutamic acid to form para-amino benzoyl glutamic acid. PABA reacts with the glutamic acid to form para-amino benzoyl glutamic acid in presence of ATP and CoA SH. ATP, CoA SH, PABA combines with glutamic acid to form para-amino benzoyl glutamic acid. This then reacts with pterin. To produce pteroyl monoglutamic acid. Pterin to form pteroyl monoglutamic acid or pteroyl glutamic acid or folic acid. This is the formation of folic acid. PABA first reacts with the glutamic acid to form para-amino-benzoyl glutamic acid. This one then reacts with the pterin to produce Pteroyl Glutamic Acid or Pteroyl Glutamic Acid or Folic Acid. Pterin moiety is derived from guanosine. From guanosine, this reaction. Next is effect of drugs. Effect of drugs. Effect of drugs. Sulfonamide drugs and antibiotics inhibits their growth by blocking the incorporation of this PABA in the synthetic pathway. That is by competitive inhibition. Sulfonamide drugs and antibiotics, they blocking the incorporation of PABA in the synthetic pathway, that is by competitive inhibition. Then higher animals including human beings cannot synthesize folic acid, so it has to be supplied in the diet. In human beings, intestinal bacteria can synthesize the vitamin and it is a good source. So in human beings, the sources are from dietary source and from the intestinal bacterial flora synthesis. That is the biosynthesis of vitamin B9. Next one is metabolism. Absorption. Absorption occurs along the whole length of mucosa of small intestine. Polyglutamate ingested in diet. Polyglutamates that is ingested in diet are converted to monoglutamates and dihydrofolates are converted to the tetrahydrofolate. This is by folate reductase. These tetrahydrofolates are then converted to methyl tetrahydrofolates which enter the portal blood and then carried to liver. So dihydrofolate is converted to tetrahydrofolate by the enzyme reductase. Tetrahydrofolate is then converted to methyl tetrahydrofolate which is then enter in the portal blood and then carried to liver. That is the absorption. Then coming to transport, it is transported in blood as methyl tetrahydrofolate. Methyl tetrahydrofolate, methyl FH4, and it is bound to a specific protein. Plasma level is about 3 to 21 Ng per ml. Coming to excretion, in urine it is about 2 to 5 μg per day. Then in feces it is 20%. In this 20% of the ingested folates that remains unabsorbed. In feces 20% is unabsorbed folates. Then 60 to 90 μg in the bile that is not absorbed, reabsorbed. Then some unabsorbed synthesis of folate by bacterial flora of intestine. Unabsorbed synthesis of folate by intestinal bacterial flora. That is this excretion in feces. Then you will see the RB cells folate. RB cells folate. This red cell folate is a reliable indicator of folate status of the body. Red cell folate is a reliable indicator. Average level is about 300 Ng per ml of whole blood on a PCV of 45%. The range is about 160 to 640 Ng per ml. That is the RB cells folate. This is a reliable indicator of folate status of the body. Next one is food sources. Food sources. It is widely distributed in nature. It is present in animal and plant tissues and in microorganisms. It is abundant in liver, yeast, kidney and green leafy vegetables, spinach and cauliflower, then meat, fish, wheat. Then fair sources are found in milk and fruits. That is the food sources. Next one is metabolic roles. Metabolic roles. That is one carbon metabolism. That is a very important function, one carbon metabolism. The folic acid coenzymes are concerned with metabolic reactions involving the transfer and utilization of one carbon moiety. Folic acid coenzymes are used for the metabolic reactions involved in the transfer and utilization of one carbon moiety, that is C1. This one carbon moiety may be either methyl, that is methyl, or formyl or formate. These are the one carbon moieties. Then 4 amino group, that is CHNH, then hydroxymethyl, that is CH2OH. These are metabolically interconvertible and they are catalyzed by an NADP plus dependent hydroxymethyl dehydrogenase. These are interconvertible and are catalyzed by the NAD plus dependent hydroxymethyl dehydrogenase, that is CH2OH, that is hydroxymethyl, that is converted to CHO. CHO means formyl, that is NAD plus NADP plus NADP H plus H plus. Here hydroxymethyl dehydrogenase. Hydroxymethyl dehydrogenase. This is CHO or formyl is converted to COOH. Same enzyme, that is H2O is released. Here NAD plus NADH plus H plus. NAD plus NADH plus H plus. So these are interconvertible. Then this F10 tetrahydrofolate, F10 tetrahydrofolate or F510 tetrahydrofolate, F510 tetrahydrofolate. These are the active forms of folic acid coenzymes in metabolism, F10 and F510. But this F5 is inactive in metabolism. That is the metabolic roles. Next we will see the clinical significance. Clinical significance of vitamin B9. First one is folic acid antagonist. We will see the folic acid antagonist. On the basis of their ability to inhibit cell division and multiplication, antagonists are used in treatment of conditions where there is unrestricted cell growth. For example, in leukemias, in erythemias and in malignant growth. So, 4 types of antagonists are there. First one is aminopterin. Aminopterin. In this, NH2 group is substituted for the OH group. OH group is replaced by NH2 group in position 4 of pyridine nucleus. Chemically, it is 4-aminofolate. 4-aminofolate. It has maximal inhibitory action, that is aminopterin. Next one is amithopterin. Amithopterin or methotrexate. Methotrexate. It is 4-amino 10-methyl folate. 4-amino 10-methyl folate. This methotrexate inhibits dihydrofolate reductase enzyme. Dihydrofolate reductase. It inhibits the methotrexate inhibits the enzyme dihydrofolate reductase and is used as an anti-cancer drug. That is amithopterin. Next one is trimethoprim. Trimethoprim or Septran. Septran. It inhibits dihydrofolate reductase. So the formation of tetrahydrofolate is reduced. This is used as antibacterial agent. Next one is pyrimethamine. Pyrimethamine. It is used as anti-malarial drug. It also inhibits the enzyme dihydrofolate reductase. So most of them inhibit the enzyme dihydrofolate reductase. So this tetrahydrofolate formation is reduced. Next one is, these are the folic acid antagonists. Next one is deficiency manifestations. Deficiency. First one is macrocytic type of anemia. Clinical aspects. Macrocytic type of anemia. In this, first one is bone marrow. Bone marrow shows arrested development of all elements, erythroid, myeloid and thrombocytes. Then megaloblasts and myeloblasts accumulate at the expense of more mature cells. That is erythroblasts and normoblasts and myeloblasts. The number of megakaryocytes decreases in the bone marrow. Then next one is in the peripheral blood picture. Peripheral blood. In the peripheral blood picture, it's characterized by a macrocytic type of anemia, then granulocytopenia and thrombocytopenia. This is depending on the degree of deficiency of the enzyme. Next one is degree of deficiency of the vitamin. Next one is clinical conditions. Clinical conditions. On the basis of prompt response to specific replacement therapy, some clinical conditions are there in folic acid deficiency. They are, first one is nutritional macrocytic anemia. Macrocytic anemia. As the name indicates, it is because of dietary deficiency of folic acid. That is nutritional macrocytic anemia. Next one is megaloblastic anemia of infancy. This is also due to dietary deficiency. Next one is congenital or inherited type. That is due to reductase deficiency, enzyme reductase deficiency. Next one is megaloblastic anemia of pregnancy. Next one is macrocytic anemia in liver disease. Macrocytic anemia in liver disease, it is due to inadequate storage or conversion. Next one is megaloblastic anemia in celiac disease and sprue. That is due to inadequate absorption. In this, there is B12 deficiency is also there. In addition to this B9 deficiency, there is also B12 deficiency also there associated with neurological manifestations. Next one is macrocytic anemia after extensive intestinal resection. This is due to inadequate absorption. So, how we will detect these folate deficiencies? So, there is a test that is known as FIGLU test. This test is used to detect this folate deficiency. That is, we will see the reaction. N4-immunoglutamate. N4-immunoglutamate is converted to glutamic acid by the enzyme glutamate 4-immunotransferase. Glutamate 4-immunotransferase. Here FH4 is converted to N5-formyl FH4. FH4 N5-formyl FH4 in folic acid deficiency. That is, this FIGLU test is used to detect folate deficiency. It is in the metabolism of histidine. Metabolism of histidine. There is a folic acid dependent step. There is a folic acid dependent step in histidine metabolism, where this 4-immunoglutamic acid is converted to glutamic acid. 4-immunoglutamate is converted to glutamic acid. This is in histidine metabolism. There is a folic acid dependent step, that is 4-immunoglutamic acid is converted to this glutamic acid. 4-immunoglutamic acid is called FIGLU. FIGLU, 4-immunoglutamic acid, that is FIGLU. Then in folic acid deficient patients, this reaction cannot occur. In folic acid deficiency, this reaction cannot occur, so glutamic acid is not formed and N4-immunoglutamate or FIGLU is accumulated in blood and it is excreted in urine. This FIGLU excretion in urine is an index of folic acid deficiency. So, accumulation of N4-immunoglutamate and in blood and it is excreted in urine. So, excretion of FIGLU in urine is an index of folic acid deficiency. Then, when a loading dose of histidine is given, the excretion of FIGLU in urine is increased further. That is called as histidine loading test. Histidine loading test. So we can increase the excretion of FIGLU. When a loading dose of histidine is given, excretion of FIGLU in urine is increased further. That is histidine loading test. That is FIGLU test. This is used for the detection of folic acid deficiency. Next one is daily requirement. Daily requirement for adults, it is about 400 to 500 μg daily. Then in infants, it is about 50 μg daily. In children, it is 100 to 300 μg. Then this requirement increase in pregnancy and in lactation. We will see the risk of excess of folic acid. These are the daily requirements. If excess is there, there are risk. So, this is the risk of excess of folic acid. Dosage over 1 mg may cause aggravation of vitamin B12 deficiency and may cause irreversible nerve damage. So, dosage over 1 mg, it causes the aggravation of vitamin B12 deficiency, and this causes irreversible nerve damage. There will be antagonism between folic acid and the anticonvulsants used in the treatment of epilepsy. Then solubility of folic acid will be low. So, large doses of folic acid if given parenterally, there is risk of crystallization in kidney tubules. It leads to renal damage. These are the risks of excess of folic acid. So, this is all about vitamin B9 or folic acid group. This is today's topic. Thank you for watching.